Stainless steel reactors are still the most preferred option for large scale, such as 5,000 to 10,000 liters, but single-use bioreactors are quickly becoming the preferred option for small-scale reactors, such as 1,000, 2,000 liter.
The choice of the bioreactor is also important with regard to the future. Will the bioreactor be able to meet the demands of future processes, higher cell densities, higher production quantities, and increased regulatory requirements?
We needed single-use equipment that equaled or bettered stainless steel bioreactors in terms of consistency and reliability, reproducibility, robustness, and most importantly, flexibility. The single-use reactors, they also need to have optimal mixing capabilities, optimal mass transfer, and very importantly for us, flexible sparging options. Our programs include our proprietary Apollo expression platform, and that combined with our Saturn MAB platform, but we also do a significant number of client-specified projects, and adoption of single-use technology has allowed us to continue to be flexible enough to meet all of these varied needs.
The choice of the automation and control systems are critical to the success of the project. The right pairing can result in many benefits, such as standardization of equipment, standardization of intervals for sampling and time data collection, validation, and also training and maintenance. Automation can also reduce the amount of human interaction with the process and the chance for human error. This leads to an overall improved level of quality assurance and compliance of the operating process when, again, the right pairing systems of automation and control are brought together. The family approach is the way to duplicate your production lines, but you only have to validate the first production line and don't have to repeat that validation work on additional identical production lines. We can exploit this family approach in single-use manufacturing facilities because the bag in the bioreactor is the same bag in additional production lines that we add to increase capacity. In this way, we can reduce the amount of work to validate additional capacity as we expand our facility.
We had the system in our facility in North America, and by analysis of that and its introduction into the UK, we reckoned we could save about 3 months in installation time, and that turned out to be the case. It was clear from the experience that they had in RTP that the tech transfer would be facilitated by the fact there was so much knowledge in the organisation there of how to use the single-use bioreactors, and also that gave us a way of managing risk of introduction of the new technology.
The scale-down ratio of a bioreactor means the difference between the maximum operating volume and the minimum operating volume. The larger that turn-down factor has a number of benefits. Number one is we can reduce the size of the inoculum train because the inoculum is much smaller for the starting volume in the production reactor. Number two, that reduces the number of vessels that we need to buy. Number three, that reduces the size of the facility that we need to build. Number four, that reduces the number of bags that we consume per batch. And number five, that reduces the overall operating and capital cost expenditures for the facility.
So the scale down is important because it allows us to have a lot of varied options for scale down modeling, but it also allows us to have a lot of varied options for scaling up. Having the process run in a vessel that's specific for your output needs is very beneficial to help minimize waste.
We looked very closely at the capacity of the assets in RTP. They had a range from small scale through to 1,000 litre, and then they also added in 2014 the 2,000 litre bioreactor. This gave them the ability to serve customers right the way through the clinical cycle. Having looked at that, we thought, well, this is probably a good idea to emulate it, have a mirror image if you like, because that would give us the ability to build on the experience they had and also to flex by moving customers from one site to another as assets became free, as well as serving the full clinical cycle.
In summary, the take-home message is that whatever technology platform you choose, it should have the performance, reliability, flexibility, and robustness to meet the demands of your processes today and the processes of your future.